a toxin that is extremely carcinogenic, mutagenic, and easy to cause acute poisoning. Its existence, although so tiny that it is difficult to detect with the naked eye, carries a toxic force that can threaten human health and even life. Among these, aflatoxin B1 is the most common form. Long-term ingestions of AF-B1 in small doses in humans will lead to embryonic malformations, mutations, and induction of primary cancer, etc. AF-B1 can also be left in animal products to further endanger human health. Aflatoxin B1 is stable to heat and is really difficult to destroy structure at cooking temperature. So even after cooking, the toxin remains in food and enters our body. The contamination caused by this is a global problem, but it is even more severe in warm, humid climates such as Asia and Africa. According to available research and statistics, approximately 4.5 billion people worldwide are potentially affected by aflatoxins. Our project aims to degrade aflatoxin B1 to reduce the problem of food contamination caused by AF-B1 worldwide. We will achieve this through synthetic biology using lacase and a set of finely controlled devices. This method is environmentally friendly and more specific, leaves no harmful residues, and preserves the nutritional value of the food, degrading aflatoxin B1 to harmless substances. The implementation of the project will bring significant environmental and health benefits. By reducing the amount of aflatoxin B1 that may be present in food, we are not only safeguarding people's health, but also reducing agricultural losses due to the toxin contamination, thereby supporting food safety and agricultural sustainability. In addition, our research will explore the application prospects of these biocatalysts, including potential uses in food processing, storage, and the feed industry. We fervently hope that this project can contribute to global food security and public health.
We have learned before that you use engineered bacteria to transport plasmids that perform their functions into the environment. Could you please tell me more about that?
Sure. We chose LGG, a probiotic commonly used in dairy fermentation, as our chassis. This bacterium is gram-positive, and its peptidoglycan layer can physically absorb up to 60% of aflatoxins in the environment, achieving a concentration effect.
To better break it down, we introduced the gene of Codeco to the strain to enable the bacterium to degrade aflatoxins completely.
I see. LGG was chosen because of its ability to enrich toxins. Could you please introduce the relevant parts of LGG in detail?
Let's go to the lab and find the team leader.
We selected the T7 promoter to control lacase expression. This puts lacase under the control of an engineered polymerase with customized inputs. Then we chose the nanobody that is a naturally occurring antibody in alpaca peripheral blood. We selected the G8 nanobody that can specifically recognize and bind aflatoxin B1. We combine this nanobody with GLUC to form a fusion protein. Once it binds to AF-B1, the GLUC is activated, oxidizing the substrate and producing a blue fluorescence. The plasmid PGG, which is G8 GLUC fusion plasmid, is used to detect AF-B1 in the environment, generating a blue fluorescent signal, which acts as a trigger for the next step in the reaction.
Additionally, we selected the photosensitive protein VVD as the blue fluorescence receptor, which dimerises upon activation by blue light. To achieve process regulation of locase expression, we split the T7 RNA polymerase coding sequence into two parts, each expressed alongside VVD. After receiving blue light, the photosensitive protein forms a homodimer, and the T7 RNA polymerase domains bind to regain activity. The plasmid PVVD, which is VVD T7 RNA polymerase fusion system, expresses two T7 RNA polymerase fragments and VVD proteins to sense external stimuli and regulate locase expression.
So, why did you choose light as a signal? Does it do any good?
Because our detection substances usually appear in grain, oil, feed, and other related products, we can use light without additional introduction of relevant chemical signal molecules to minimize the impact on the detection of substances.
Furthermore, to prevent the escape of the engineered strain into the environment and avoid contamination, we selected a glucose-inducible suicide system. We observed that glucose concentrations vary between feed, grains, oils, and the gut of the higher mammals. compared to the external environment. Therefore, we considered using a promoter, PGLU, that can respond to changes in glucose concentration. We aim to prevent the escape of any bacterial contents upon cell death to avoid any potential risks. Thus, we chose to use the MAZF gene, which encodes an endoribonuclease that cleaves RNA at ACA sites, leading to microbial death without causing bacterial cell lysis. The PGLU promoter controls the expression of the MAZF, and when glucose concentration is low, the suicide gene is expressed, inducing cell death.
I see. I finally understand the principle of the project. So, may I ask how you verify the principle using experiments?
Sure.
In the design workflow, the key genes will be synthesized, and the fragment will be connected to the vector using either infusion or T4 DNA ligarase. After transformation and selection, a complete expression vector is obtained. After the large-scale culture of E. coli, enough plasmids are extracted for subsequent experiments. We first measured the growth curve of LGG as the chassis organism. We set up 3 parallel experiments and manually measured OD600 every hour for 16 hours after inoculation. The collected data was fitted to a growth curve, proving that our chassis organism could grow normally.
Based on literature, we used 2 different competent cell preparation and heat shock transformation methods for 2 activated strains to attempt to transform PCG into the engineered strain. However, after initial screening using contamination resistance and verification by colony PCR, the first transformation results were not ideal.
We then adjusted our strategy and used electroporation to simultaneously introduce PCG and PVD plasmids into the chassis organism. After preliminary screening using spectinomycin and kanamycin, we selected the surviving single colonies and conducted colony PCR. The results from the gel analysis showed that the fragment length of the samples matched our original design, indicating successful transformation with satisfactory efficiency.
We selected the cleanest transformants for further purification and culture to later introduce the remaining 2 plasmids.
Alright, how about the results of your experiment?
Let's ask the members in the experimental group.
Before constructing the complete engineered strain, we first verified the suicide function of PBS. We selected LGG without the suicide plasmid as the control group and set up 10 experimental groups with different glucose concentrations, repeating the experiment 3 times for each concentration. After the bacteria grew to the logarithmic phase, we inoculated 5 microliters of the bacterial solution into 5 milliliters of MRS medium with different glucose concentrations. The tubes were then placed in a 37° centigrade incubator for 12 hours and OD600 was measured.
Compared to the control group, LGG transformed with the glucose-sensitive suicide plasmid PBS-MAZF showed significant growth inhibition. Particularly at lower glucose concentrations, the inhibition effect was more pronounced, leading to a marked reduction in LGG growth and reproduction rates. Within the 0.1 to 1.0 concentration range, we observed inhibition rates exceeding 80%, while at low concentrations, the inhibition rate approached 100%. Based on these findings, we conclude that in vitro, the glucose-sensitive suicide switch can function effectively when glucose is absent or in very low concentrations in the fermentation environment.
In addition to the theoretical and experimental operation of the project, we also pay attention to the significance of our project to society. We believe that our projects should not only stop at the exploration of knowledge, but also provides practical measures to address real-world issues and fulfill the needs of the people. This is why human practice is particularly important, as it gives our research a course. Our human practice is divided into 4 parts: market research, public survey, interview research, and communication with other IGM teams.
In the market research, we learned that the degradation of aflatoxin B1 by the current relatively mature physical-chemical methods may cause risks such as secondary pollution and nutrient loss. There is an urgent need for efficient, safe, and side effect-free detoxification technology.
In the public survey, we conducted online surveys for different groups, including their perceptions of aflatoxins, synthetic biology perceptions, and product design recommendations. The survey was conducted among 173 members of the community, most of whom had a bachelor's degree or above, who understood the danger of the aflatoxins and recognized the potential of synthetic biology to degrade toxins. The result of the survey shows that the majority of people support the application of engineered bacteria, and a few people are resistant to genetically engineered products. This indicates that the future project needs to further explore issues for public interest.
During the interview, we discussed with experts from DSM, China Chamber of Commerce for import and export of foodstuffs, native produce and animal husbandry, Chinese Academy of Agricultural Science, Shandong Customs Grain and Oil Testing, Sichuan Quanwei Food, and Kofco Shanhui peanut products. The experts emphasized the importance of controlling microbiotoxin contamination to ensure food safety and introduced international monitoring standards and methods. Experts said that from a technical point of view, the project is significantly innovative and feasible, and it's expected to provide a new solution to solve the problem of aflatoxin B1 degradation. At the same time, the experts provided valuable advices on marketing of the technology and suggested that the project should focus on agricultural feeds, especially silage, rather than dairy products, as dairy products are required to be destroyed as soon as aflatoxins are found to be present in the Codex Alimentarius Commission.
This provides an important reference for our subsequent R&D and market planning. Through these exchanges and interviews, the team members not only deepened their understanding of the current situation of the feed and grain and oil industry and aflatoxin treatment technology, but also modified the application direction of the project on this basis, injecting impetus and confidence into the future development of the project.
On September 21st and 30th, under the organization of the IGN team of Beijing Institute of Technology, we conducted in-depth synthetic biology exchanges and populate outreach, improved experimental design, and learned from several teams, like the teams of high school affiliated to Renmin University, Beijing No. 11 School, and Beijing Institute of Technology. We explored the application of the paradigm of synthetic biology in the project, and how high school students can contribute to the field.
To ensure safety and environmental friendliness, we implement necessary precautions to mitigate harm. Our safety measures encompass chassis, parts, suicide system, and laboratory safety. We utilize LGG ATCC 7469, a low-risk engineered strain, for program safety. Prior to plasmid construction, we verified the main gene source against the IGEM whitelist. The PGLU promoter regulates the MAZF suicide gene, activating under low glucose conditions. All team members adhere to laboratory safety protocols, including hygiene, attire, eye protection, glove use, and material handling.
Next, we plan to measure the expression levels of PBS-MAZF in the LGG engineered strain, both mRNA and protein levels, and assess the efficiency of this suicide system in environments containing alternative carbon sources. We will then use the existing FB1 detection kit and a microplate reader to evaluate the sensitivity and degradation efficiency of the crusher system, which successfully expresses 4 key components against AFB1. Additionally, we will investigate the performance of our biological components under various environmental conditions, striving to improve their sensitivity, efficiency, and safety. Our goal is to develop a probiotic product suitable for the grain and feed industries that can effectively remove AFB1. At the same time, we will continue to optimize and expand our fine-tuned regulation system, as well as establish a nanobody library to enhance its applicability and flexibility in a broader range of fields.